Welcome to a tour of the Nobel Prize Factory. This is the MRC Laboratory of Molecular Biology, or LMB, a world-class research institute in Cambridge. At the LMB, we study fundamental questions of biology at the molecular level, with the goal of using this knowledge to tackle major problems in health and disease. Today, you'll get to see where and how we do this.
Hello, good afternoon, and welcome to our very first live virtual tour of the Laboratory of Molecular Biology, or LMB. My name is Shahana Ahmed. I've been working in scientific research for over 20 years, and currently at the LMB, I'm involved in mutation detection. Today, I've hung up my lab coat and I'll be one of your tour guides. Before we get started, I just want to quickly go over the practicalities of what we're doing and how things will work. Some of my colleagues from across the building will be showing you inside their labs and giving you an insight into the kind of work that they do. We'll be running a live experiment, fingers crossed, and there'll be a quiz. To take part in the quiz, you can just watch the video and answer the questions for yourself. But if you're feeling super competitive, you can compete with others by clicking on the link that we've pasted at the bottom of the YouTube video, or by using Slido on a separate device and typing in the code LMB Tour. So if that's what you're planning to do, make sure you have that second device to hand. At the end, there'll be a live question and answer session. So if you have any questions at all throughout the tour, just pop them in the chat next to the video and we'll make sure we'll try and answer as many as we can. Now, I think that's all the housekeeping done. Now let's get started with the tour of the Nobel Prize Factory. So the LMB has been dubbed the Nobel Prize Factory because of the number of awards that the LMB researchers received in the last maybe 70 years or so. However, it's really important to point out that science doesn't really work like a factory, and scientific breakthroughs can't be precisely planned or manufactured. Our main goal is advancement of scientific knowledge, and any prizes as a result are a much appreciated bonus. So the LMB scientists, support staff, and technicians have worked really hard and made some important major breakthroughs. We can see some of these on our timeline. So our history dates back to 1947. As you've probably realized, this fabulous shiny building is much newer. We've been here since 2013. So this banner shows all our important discoveries. For example, our work on monoclonal antibodies. In one form or another, monoclonal antibodies have been important for the treatment of a staggering number of diseases. And then there was a discovery of DNA sequencing technologies. These have been important for the Human Genome Project. I was fortunate enough to have been involved in the Human Genome Project in the 1990s. Nowadays, you might associate DNA sequencing with maybe tracing your ancestry. There's also been lots of groundbreaking work done to highlight the structure of proteins. I can't go through all of them now, but do check out our website to see more. Now let's move on. On the other side of this corridor is our structural studies division. Here, our scientists are developing novel techniques to understand biology at the level of the atom and are looking into the precise 3D structure of proteins. It's important to study proteins because proteins can act as molecular machines and enzymes. They can help a cell form its shape. Proteins can attach to the surface of cells and allow interaction between them. There's not really much in our bodies that proteins don't do. But to have the ability to study them at the level of the atom means that we can figure out how they do all this multitasking. And probably more importantly, what would happen if these proteins go wrong and how could we potentially fix them? We're now going to go into the structural studies labs and meet John Long and Christina, who are going to tell you more about this work.
Hi, I am Zunlong Ke, a researcher in Structural Studies Division. We use cryo-electron microscopy to study viruses such as HIV, Ebola virus, influenza virus, and more recently, SARS coronavirus 2. SARS coronavirus 2 is the virus that causes the COVID-19 pandemic that we have right now. Cryo-electron microscopy is a tool that allows us to visualize atomic details of proteins. One of the top scientists, Richard Hansen, in this building was awarded the Nobel Prize because of his contribution to the development of the technique cryo-EM. Before we can image our sample under an electron microscope, we need to prepare our sample. The sample can be proteins or viruses that needs to be plunged further rapidly into the liquid ethane. And the liquid ethane is at negative 180 degrees Celsius, and that is really cold. After the sample is prepared, we'll load them into the electron microscope. And here is a model of an electron microscope. Now we have the source of the electrons that fires electrons through the column, and the sample is placed at this plane that will be recorded by the detector down there. Then we'll have a series of 2D projections of this protein, and we can reconstruct the three-dimensional structure of these proteins. And that method is the same principle as how the CT scans work in the hospital. Cryo-electron microscopy is such a powerful tool that it allows us to visualize SARS coronavirus 2 particles. And here are the SARS coronavirus 2 particles, that they are about 100 nanometers in diameter. And if you look closer to these particles, that you can really see the spike proteins coming out of the virus. And we can build the three-dimensional structure of the spike protein. A further analysis really allows us to see the differences on the receptor binding domain of this spike protein, and that is the region the virus uses for binding onto the cell and replicates into the cell. If we put all the information we gathered about the virus spike protein as to this shape and the positions, as well as how it tilts towards the membrane, we can recover the three-dimensional information of this virus, and we can build the three-dimensional model of it. And here are two more examples of this virus models. By studying the SARS coronavirus 2 particles, it really helps us to understand how the virus works, and it will be beneficial for the drug design and vaccine development.
Hi, I'm Christina Heroven. I'm a scientist in Radu Aricescu's lab at the Division of Neurobiology here at the LMB. And we look at neuronal receptors. So these are protein molecules in your brain that are very important because they allow the communication between neurons. So they're essential for every cognitive function you could imagine. And we want to obtain atomic structures of these neuronal receptors. That means we want to look at them at very high detail. So to the detail of an angstrom, that's like the millionth of a human hair. So super tiny. And as you've already heard from Zunlong, cryo-electron microscopy is a very powerful technique to look at viruses like he does, but also at these very tiny protein molecules that we're interested in. So today I'm going to show you my favorite microscope, which is this electron microscope Titan Cryos. And as you can see, this microscope is huge. So in very general terms, a cryo-electron microscope contains an electron gun at the very top that generates the electron beam, which is then focused by a series of lenses, and the sample is inserted somewhere around there. And at the very bottom, you would have a detector that would generate the images that are then transferred onto the computer. Given that the microscope is so big, you might be surprised to find out how small our samples are. So these are examples of cryo-EM grids discarded by previous users because they've been used already, and they're these small, about 3 millimeter big grids that we need to put into a cassette in order to put it into the microscope. And that cassette looks something about like this. So there are 12 slots in there for 12 grids. And then we have to transfer this cassette into the microscope using a container with liquid nitrogen. We're very lucky to be doing cryo-EM at the LMB because it is still a developing field and we have people in the building constantly working to improve the hardware of the microscopes, but also the software needed for data processing downstream, so that we can not only always improve the quality of the data we obtain, but also make the most out of it.
Hello. Hello, my name is Yulia. I'm a second year PhD student here at the LMB in the cell biology division in Liz Miller's group. And you've joined me here on level 3. And I guess the good thing of being on a virtual tour is that you didn't have to go all the way up these steps. And there's more of them than it might seem, because we also have these interstitial floors where all the pipes and wiring lie to keep the building running. And now let's go to Stephen for the lab experiment.
Good afternoon, I'm Stephen, and I'm part of a small team that runs a biophysics facility here in the LMB. So as the name suggests, biophysics uses physical principles to understand biological questions. So we measure mass, size, the strength of binding between biomolecules, how quickly they come together, and how they fall apart. And you can imagine that these techniques are applicable to lots of areas of research within the LMB, but today I'm only going to show you just one example. So a recent project we've been involved in is been on the protein ATM. It's got an important role in the cell when DNA becomes damaged, especially at sites where both the double strands become broken. You can imagine that this would be catastrophic for the cell, because all that information is going to be lost. So ATM raises the alarm in the cell, stops cell division, and rallies the repair machinery to those sites of damage. We really want to understand how it works, so we can understand also what goes wrong when the repair machinery fails, which can lead to diseases like cancer. Now, we find that ATM doesn't do its job alone. There are a group of proteins that come together to recruit it to these sites of DNA damage. And we want to understand what the structure of that is. And the first way of doing that is to understand, how this machine is put together. It's like Lego blocks coming together. And so we want to see, what is joined to what. And so one way we can do is we can count the individual Lego blocks and to see how they're coming together, and we can do that by measuring their mass. Now, to do that normally in solution, we'd need a special blacked out room with lots of lasers and mirrors, but we've got this new instrument here where all that's been miniaturized in this little black box. And what it's going to do is we're going to measure the mass of a protein using light. So let's have a look and see how it's going to do that. So if we open it up here, we've just got a glass slide here, and a laser is going to be fired up from the instrument, and it's going to hit that glass slide and then be reflected back down through a microscope and into a camera. So if a protein lands on that glass slide, what will happen is that protein will scatter some of the light, and that will interfere with the reflected light coming back down to the detector. And the type of interference we get is called destructive interference, and what you see is black spots, as you can see here in this image here. And how dark these black spots are is a measure of the mass of the protein. Right, so let's do the experiment. Let's see how many of these proteins are going to join together. So I'm just going to pipette some of the solution onto the glass slide. And as you can see, the image is quite messy. And that's because there's all these imperfections and scratches on that slide. And so what we're going to do is we're going to take a movie. Because all this background is going to be the same all the way through the movie, if we take different frames and look at the differences, then we can see the black spots appearing. So I'm now going to record the data, and we're going to have to wait a bit of time to record and for the computer to analyze the results. So if you've got any questions, then please put them in comments below this video here, and come back later and we'll see the results and see how many of the proteins have joined together. I'll see you later. Bye-bye.
Hello, it's good to see so many of you watching all the way across the UK and everywhere in between, Germany, Italy, and Colombia. That's great. And as Steven said, we'll be back with him later, where he will show you the results and answer any questions that you have, which you can type in the chat next to the video or on Slido. And we're really keen to answer your questions, so do type them in. And before we start the Q&A, I still have some labs for you to show, so follow me to the cell biology division. So we're now in the cell biology division, well, actually my bench, which is conveniently located right next to the write-up area. And much of the research done within this division and LNB in general is focused on the very fundamental questions and processes that happen within our cells, such as how is genetic material read and translated into protein, how are proteins translocated across the cell, and how do cells and tissues take shape, just to name a few. And in the lab that I'm in, Liz Miller's lab, we're specifically interested in the question of how are proteins transported outside of the cell, and in particular, how are they recruited into the secretory pathway at the very earliest stage at the endoplasmic reticulum. And so to tackle these fundamental questions, scientists employ a lot of model organisms, which vary from bacteria to yeast, even archaea, worms, or flies. And speaking of flies, now let's go to animals.
Hi, I'm Annabelle, a third-year PhD student here in cell biology in Katia Roper's lab, and in Katia's lab, a number of us work on fruit flies. Fruit flies are a model organism, and we've been working on them for around about 100 years now, and because we've been working on them so long, we have a number of genetic tips and tricks available to us to help answer the biological questions that we have. One cool trick that we use is we can express proteins not normally found in flies and express them inside the flies and their embryos. One example of this that we use extensively in the lab is to express a protein normally found in jellyfish called green fluorescent protein, or GFP, and we can direct the expression of this protein inside flies. The flies I have here don't look anything particularly special, but if you put them under the microscope and illuminate them with the right laser, their eyes go from their normal white colour to fluorescing bright green. We have plenty of other examples of this in the lab, including an example that I use in my PhD project, where I express this green protein inside the salivary gland of fruit fly embryos. And the reason I work in the salivary gland of fruit fly embryos is that I'm interested in how tubes form, and the salivary gland and its development is a very simple example of how a flat sheet of cells can turn into a 3D tube structure. I tag these cells with GFP, and using machines found here in the building, I can separate embryo cells that are green from non-green. I can then take the collection of green cells that I have and do an experiment called single cell sequencing, where I can identify the genes that are expressed inside this patch of green cells right before tubes begin to form. Once I have a list of all the genes that are expressed during this time period, I can start to look for the genes responsible for how tubes do begin to form. Why we need to know what genes help tubes form correctly is that in development, when we're developing as an embryo, there are lots and lots of tubes that need to form in a correct manner, and understanding how tubes form in a correct manner can help inform us of what happens when tube formation goes wrong. And when tube formation goes wrong, it can lead to a number of biological conditions. Here in the building, we are not the only people working on flies. There are lots of other questions being asked and helped being answered using fruit flies, including how proteins are transported from one side of the cell to the other, how those proteins are packaged, and over in neurobiology, people are working on how neural circuits combine to control complex cell behaviors. Thank you so much for dropping by Cell Biology. It is bye from me, and it is bye from the flies.
Here on the top floor is our canteen, where the LMP staff and students come for lunch or for tea and coffee breaks throughout the day. Of course, like restaurants everywhere, it's looking a bit different in the time of COVID, but it's played an important role in the life of the lab and of the Institute. Since the founding of the LMB, our researchers have come up here to the canteen on the top floor for tea, coffee, and lunch breaks so they can get together to discuss their work and even help each other to solve any problems they've been facing in the lab. In fact, these mid-morning and mid-afternoon breaks for tea and coffee have sometimes been given as an explanation for the great success of our scientists working here. So naturally, the canteen has also been the place where we have celebrated those successes, and a number of Nobel celebrations have taken place in this canteen, both in our building and in the old one. When the weather is nice, actually, we might take these breaks outside, where we can see some of our neighbors on the Cambridge Biomedical campus, one of the largest in the world. Being right next door to the hospitals and other research organizations allows easy collaboration with medical doctors, as some research at the LMB is more closely related to health and disease than a lot of the work we've shown you so far. For example, some of our researchers have worked with samples from patients with inflammatory bowel disease, ulcerative colitis, patients with a variety of kidney diseases, TB infections, and more recently work with live SARS-CoV-2, the virus behind the current pandemic.
Hi, my name is Anna, and today I'm going to show you around our containment level 3 facility where we work with SARS-CoV-2, which is the virus responsible for COVID-19. Access to this laboratory is restricted to very few people, and they all need a special training to be allowed in. So far, only Charlotte and myself had the training to be allowed to work with SARS-CoV-2. The first thing to do when we enter this laboratory is to put our PPE. That comprises of gown, apron, and first pair of gloves. Later, when we're ready to start our experiments inside the safety cabinet, we can put our oversleeves and second pair of gloves. So the safety cabinet is keeping us safe from all the aerosols generated during our work. This is by aspirating all the air from inside and from outside of the cabinet, as you can see here on this demonstration with dry ice and water. The plug assay is one of the assays I use quite a lot. It's a really easy, simple method to figure out how many viruses we've got in our preparation. We dilute the virus and then add it to cells, and after a few days look at the virus spread. But because we add the agarose, which is like a little jelly, into the cells, the virus can only spread to the neighboring cells. So it kills the cell in the middle and then spread and also kills the cells around. So when we add the dye, we can see where the virus landed in the first instance. That's forms a little hole that is not blue. And in this way, we can really easily count how many viruses we had initially. We can use this assay to verify how many viruses we've got in our solution, but we can also compare different conditions. For example, use some antiviral treatment, and then after a couple of days collect the samples and by plug assay verify if we produced less viruses when antiviral drug was present. So when we're done with the experiment, we have to clean and decontaminate everything. For that, we spray all the items thoroughly with disinfectant. And all the items that we used are put in a contain waste jars. That also includes our oversleeves and outer pair of gloves, which are not allowed to leave the safety cabinet. The projects we are interested in vary from basic biology, understanding of the virus, immune response to the virus, but also viral therapeutics, antiviral therapeutics, and new vaccine strategies. So our favorite protein is nucleoprotein, which is actually hidden inside the coronavirus. And we are looking into if the immune response to this protein is in any way useful to fight the virus, and whether it would be beneficial to add this protein to the future vaccines. So for the moment, all the available vaccines contain spike protein. So we would like to know whether adding nucleoprotein to spike vaccine would be helpful, especially that nucleoprotein is stable and it doesn't mutate as often as spike. So that could help against these new variants that are developing all around the world.
Alright, it's quiz time now, so let's take the lift back down to the ground floor and I'll see you in just a minute. Hey everybody, hope you've enjoyed the tour so far. It's an absolutely beautiful building to work in and to live in, as is Cambridge. Just as a quick tip, if you have any questions about anything that you've seen so far, please put them in the chat or in Slido so we can discuss those in the Q&A. Since we can't do any live questions later, put them in the chat now. Anything about science, about Cambridge, about the LMB, any question will be welcome. So my name's David, and now you're in the molecular immunity wing here at the LMB. We also go by the nickname the Uni Wing because the researchers that are found in this wing are all from the University of Cambridge, and we kind of use this as our home to do research. What's special about the molecular immunity or the Uni Wing is that we work really closely with folks at the hospital in Addenbrooke's and in different hospitals across the UK, looking at research that is of translational purpose, which means that it has a lot to do with clinical medicine and with patients. And so we do a lot of human work to look at human diseases as well as animal models, as was shown before. I'm a first-year PhD student. That means that I have finished my undergraduate degree, and I'm now studying to a particular scientific question that interests me and my lab. And at the end of it, I will be a doctor. So we will now start the quiz that all of you should be able to have access to. So if you look on the YouTube link, there should be a link at the bottom of the description. If you click on that, it should open Slido, which should look something like this. And then you can put in questions and you'll answer them. Take a look. Something like this. Cool. So we'll get started with a quiz. If you're not sure about any of the answers, just give it a wild guess and it'll be fun. We'll see who the winner is.
Hello, I'm a researcher at the LMB and I use microscopes to investigate Staphylococcus aureus infection of immune cells. And Staphylococcus aureus is a bacterium that can, for example, infect the skin, the heart, or the lung. And with this microscope, I can really zoom in and take small movies of the bacteria inside the immune cell. And here you can see an example movie of the bacterium, which is in blue at the beginning, and when the infection was successful, then the bacteria turned green. However, not all Staphylococcus aureus can cause a successful infection. So what do you think? What percentage of humans carry Staphylococcus aureus in their nose without the bacterium causing a successful infection? Is it 5%, 10%, 30%, or 100%?
Great. So that was Sina. And just to rehash your question, she asked what percentage of people carry Staphylococcus in their nose or on the skin even without causing a successful infection. It's looking like people are voting pretty quickly. Really good. Looks like some good answers. Excellent. Well, something quick about Staphylococcus or staph infections or staph bacteria is that they're considered commensal, which means that we have a kind of interesting relationship with them because they can be on our skin or in our bodies and be healthy, but then they have the potential to cause disease. And so that's what a commensal is. Looks like all our answers are in, and I would say the correct answer is 30%. So 30% of people, which is 1 in 3, can carry staph either on the skin or on their nose without causing an infection. Great, well done. Let's move on to the next question, which Kendra is going to introduce, and she's from the lab next door. Let's take it away.
Hi, my name is Kendra, and I'm a research associate here in Jorgo Modis' lab at the LNB. In our lab, one of the topics we study is the proteins that help the cell recognize it has been infected by a virus. We are using a combination of biochemistry and structural biology to build our understanding about the function of one of these innate immune proteins, MDA5, and its interaction with other proteins on a molecular level. As you can see here, MDA5 assembles along the length of double-stranded RNA, and we are able to visualize the resulting filaments and determine their structure using cryo-electron microscopy. My question for you is as follows. What kind of nucleic acid does a virus contain? A, deoxyribonucleic acid, or DNA, B, ribonucleic acid, or RNA, C, either DNA or RNA, or D, none of the above.
Great. So that was Kendra asking about viruses, and our most beloved virus, SARS-CoV-2, it comes to mind. And the question was, what type of genetic information do viruses have? Is it RNA, DNA, either, or none of the above? If you're not comfortable with, if you're not sure what's going on, just take a wild guess. It's all for fun anyway. Some examples of different viruses that you've heard of besides SARS-CoV-2 are measles viruses, herpes viruses, chickenpox, smallpox, and all those have genetic material that come in a few different flavors. Looks like all of our answers are coming in, and the correct answer is either. So the viruses come in lots of different flavors, as I mentioned before. They can have either DNA or RNA, and that can be expressed as single strands, which means they only have one copy of it. Well, kind of like that. They have one copy of it or two copies of it if it's double-stranded, either an RNA or DNA. All right, let's go on to the last question of the quiz and take it away to Divya.
Hi, I'm Divya Arora. I'm working as a research associate in Dr. Enders Photo Laboratory in Cambridge University at LMB. My keen interest is to understand bacterial cell infections. So for today's engagement, I'm going to share a small experiment which will be followed by a question to all of you. So I have taken some green color expressing bacteria which are used to infect human cells. After infection, I see there are two kinds of cells. The cells which cannot get infected, called as uninfected cells, and the cells which get infected and now they show green color due to the presence of green bacteria. So now I have a machine called as FACS. This machine has a laser and a detector. When I put these infected cells into this machine with the help of the laser sort these infected cells into uninfected and infected populations. This machine also gives us a plot like this, in which we can see on the x-axis, say there is an increase in the green color intensity, and on the y-axis there is an increase in the red color intensity. We can see two different populations on the x-axis. On the left of x-axis we have the uninfected population. On the right we have the increased green color expressing human cells, which is the infected population. So for my new experiment, suppose I have used the same machine FACS and I get a new plot looking like this. Can you guess that which color of bacteria I have used for my experiment? Is it a green color bacteria? A red color bacteria? Green and a red, both, and none of the above.
Welcome back. That was Divya with the last question, asking us what color of bacteria did she use for her infection experiment. The options are green, red, both, or none of the above. Looks like you guys are answering pretty quickly, and looks like we're getting the right answers, a bit of a mix. Great, you guys are doing it excellent. All right, and the correct answer, like most of you guessed, was red. Red bacteria was used in her experiment. Very well done. Hope you've enjoyed the quiz and hope you've enjoyed the tour so far. We're going to continue on with the experiment with Stephen out in biophysics. Hope you enjoy the rest of the day. Take care.
Welcome back to Biophysics. If you remember before, we were going to look at whether proteins that recruit ATM to the double strand break had joined together in a group. So since you've been away, the computer has analysed the data and it's looked at all the individual spots, calculated how dark they are, and then converted that into mass. It's also then counted up how many different spots of different masses there are to make this histogram of all the landing events. And very excitingly, I can tell you that it looks like all the proteins I put on separately have all come together in one big group here. I can tell that from the mass as well. And there's maybe some little small species here, small proteins that haven't quite joined on yet. So this is really going to help us in our bid to understand how all these proteins are joined together, and so they're going to help ATM then help the cell repair double strand breaks. I also said before the break that I was going to answer some questions, and I see we've got some time to answer them. If I don't answer your question, then please write a comment in the comment section below the YouTube video, and we'll review them after about 2 weeks and we'll try and get back to answer them. So the first question I have is, how much does this laser system cost? Well, that's a bit commercially sensitive. You'll have to ask the company. But as I said before, you know, they've taken an entire room of special optical equipment and managed to miniaturize it within this black box, and also do lots of image processing as well. Let's just say that it's above £100,000. So it's very expensive, but it does a fantastic job. So a second question here is, how can a light microscope see individual proteins? So if we just go back to the movie, and so what we're looking at here is we're actually looking at individual proteins landing on that glass surface, but we're not actually seeing the proteins like we were, say, taking a picture like in the cryo-EM. What we're looking at is we're looking at the scattering effect of those proteins So these spots are much bigger than proteins. And if, say, you wanted to look at a protein with optical microscope, then you need a technique called super resolution microscopy, where you would label a protein with something that's going to be very bright, and you have to use very, very special techniques to be able to look at proteins. And you can do that even when they're in cells. And another question here is, well, how are the darkness of the spots then related to the mass? Now, proteins are quite small and they're very, very weak scatterers of light. But what people who've developed this technique have found is because proteins are made up of the same things, whatever size they are, that the amount of scattering and hence the interference and how much darkness you get is linearly dependent upon the mass of the protein. So what we do with all the instruments, we have to calibrate them. So we put in proteins of known masses, they're very standard proteins, and of a whole range of sizes, and then we measure their darkness, and we can draw a straight line between those, so that if we get a protein where we don't know what the mass is, we can look at what the darkness is and then relate that back to our standard curve. But I see now that we're running out of time, and you've got to go downstairs to the Q&A with Shoshana. So all it leaves me to say, thanks for dropping by today, and I hope you've enjoyed it, and hopefully see you next year. Bye-bye.
Hi again. I hope you've enjoyed our tour. This is our very last stop, and we'll be doing a question and answer session with some of my colleagues who've been showing you around their labs. Hello, everybody.
Hi.
So we're going to try and answer as many questions as possible, but if we don't get to yours or you have more questions, just write them down in YouTube, and over the next weeks, we'll try and answer them. So let's get started. Are you ready?
Yes.
Right. Okay, this is, how has COVID affected your day-to-day work in the labs?
Oh, good question. So for the very first lockdown, most of us were working from home. And since then, it has been very important to coordinate work in the lab so we can do it safely and at a distance.
And I was lucky enough to work during the first lockdown, and there were literally, 3 scientists working with tons of support scientists, working together to tackle the COVID-19 problem together.
For me, I spent the first couple of weeks at home, and then we decided to open our containment level 3 lab. So then I started coming, preparing the lab, and also spending lots of time writing all the procedures, how to safely work with the virus. So that was very, very intense, but we were able to open the lab in July, and since then I was, lockdown, no lockdown, we're just in the lab all the time.
And has anybody got COVID-19 working with the virus?
No, of course not. That's, everything is just done so safely. All the procedures are in place. It's really all secured, and we all know what we're doing. It's basically more likely to get the virus in the supermarket than actually in the lab. Yes.
So Annabelle, there's a question for you. What is specific about fruit flies that means they're good for research compared to any other living organism?
Oh, okay. So there are a bunch of organisms that people like to work on called model organisms. So they're researched really well. But fruit flies have a certain number of specific advantages. One, for example, is they have a really quick turnaround. So they go from egg to fruit fly in 10 days. So you can kind of generate lots and lots of fruit flies to do large scale experiments. And also you can do plenty of different genetic manipulations on them as well. We've got lots of tools already in place. So, and they're very inexpensive as well. So that's always useful.
This is a good one. Is it difficult to be a scientist?
I think it's actually not. I mean, it's super exciting job. You know, it's every day we're doing something else. There's no monotony in it. So it's not like a job. It's like doing your passion every day. So it's really great. It's loads of fun.
Sorry, go ahead.
No, that was it. It was lots of fun.
But I guess it's not easy as well, because it's easy to not to do something, but it's difficult to do something. I think to be a scientist is that you have to be motivational, be passionate, and to keep the momentum like the whole time.
Okay, so this is actually going back to you, Zongyong. Why do you need to keep the virus so cold to study it?
Right. Why do we plunge the sample down at negative 190 degrees Celsius? It's because our imaging technique requires that the sample needs to be fixed, and the cryogen is a way to fix the virus and still keeps its structure at its very original state. So by doing that, we can take images at really high resolution but still very clear. If things are moving, then you get a blurry image. So that's why.
Okay, and I have a question for you. It's quite specific. Okay, so how do you discover different vaccines? And then going along from that, can we use membrane glycoproteins, small envelope glycoproteins, or nuclear plasmid proteins to create a vaccine? If so, why are spike proteins preferred?
Yes, so maybe I'll answer the second question first. The spike protein is the most immunogenic. We get most of the antibodies produced to this protein. And for most of the viruses that the vaccines are developed, that's the strategy is used. So I think with this pandemic, everybody just, follow along and decided to just not to do anything crazy and just follow our expertise to other viruses, just do stuff as fast as possible. In terms of how we do discover different vaccines, so there are already lots of strategies in place. We can use viral glycoprotein. We can use whole virus, which is inactivated, for example, or viral nucleic acid, like mRNA vaccines. So, and then obviously we can modify the protein in different ways. So just testing all these strategies takes a lot of time. If you have something new and you want to test it in the lab, and that's what we are doing, for example, we inject this candidate vaccine into mice, and then after some time, just get the blood and have a look if there are antibodies raised. We can also challenge these animals, meaning that we can infect them and see if they actually are protected. So that's usually the first stage of that kind of vaccine discovery. So we try with animals. If animals are protected, there's a big chance people will be protected as well. So then we can go into the next stage.
Thank you. So Christina, there's one for you. So why is your microscope so big?
Oh, that's a good question. So electron microscopes are in a way a lot more complex than light microscopes. So we need many electromagnetic lenses to focus this beam of accelerated electrons. So there are many components in there. Also a sophisticated vacuum system. So we simply need a lot of space to accommodate for all of these pieces of equipment.
And on the same question, theme of size, Annabelle, how small are your embryos, the fly embryos?
They are 50 nanometers across.
We're testing you now.
You're put me on the spot here. Basically, if you take a ruler that you have in your house, they are smaller than a centimeter. So they just kind of span that. So they're millimeters in width. No.
So this is a question for you all. Why did you become a scientist?
Okay, so I'll start trying to keep it very brief. So I come from Chile and I always grew up very close to nature. So I was fascinated by the natural world and I wanted to understand it better. So that's why I became a scientist.
I always liked biology in high school. And so I kind of just followed my interests as I went along, and ended up here pretty much.
I thought the DNA double helix was fascinating when I was in, I think, middle school. I didn't know much about it, and then just, followed into biology in college, and then I discovered my interest in viruses. So I'm here studying structures of viruses, and I think it's fascinating. And LMB is like really good place to do that.
Well, I was always interested in biology and medicine as well, and also got more into viruses after reading a book about how first vaccines were developed by Ludwig Pasteur and Edward Jenner. And also the movies like Epidemic, Dustin Hoffman, all this old school stuff is just super exciting when you're a kid. And I ended up doing something quite similar, although, don't believe in movies all the time. Sometimes it's very accurate.
So another question, how much time would you dedicate working on a research project? Do you ever go home?
Well, I'm at home now, so yes. Yeah. But so the full course of my PhD project so far is 3 years, and maybe I'll stick around for one more year. But in terms of working hours, kind of depends how long my experiments run. Most people, or some people here, work 9 to 5. Some people work a little more hours, a little less, depending on where their experiments going throughout the day.
And for like operating the really high-tech machines like Titan Crios like Christina and I do, sometimes we have to stay overnight, or if there's troubles comes, because we have dedicated time for those imaging sessions, so we have to get the most out of it.
They're very precious machines, so we want to make the most out of them.
I could just maybe add that can also do just the normal working hours. Obviously, there are some exceptions that you have to come on the weekend, but I have 2 children, and I'm still, in the lab within, the day 9 to 5 usually, and it's still going well. So you can be a scientist and you can still have a family. So don't get discouraged by the fact that, sometimes you have to go at night or on the weekend, but that's, depends the project, depends the time, and it's always your choice.
So any of you guys who are working with coronavirus, how do you make it safe, and where do you actually get it from?
So we've got our clinical strains from the hospitals. So the hospitals, or like Public Health England, they store all these things. So when we ask for something, and they can send us the strains. How make it safe? I mean, if you saw on the movie, everything is inside the cabinet. All the tubes or the plates which are infected are also always coming out of the hood in the double bags, in the plastic box. Everything is secured, everything is thrown away inside the bins, inside the cabin, and everything is disinfected. So it's just very, very strict procedure that you have to follow. And if you do that, then you're safe, basically.
And then when we put those samples into the microscope, initially, like last year in March or April, we got a bunch of batch of viruses. That's of course not infectious, fixed and not live anymore. That we got from our collaborators in Germany. And this year, because UK variant, we have obtained also inactivated virus, like the dead ones, from collaborators in London to study. And those, of course, we make sure they are not infectious anymore before we can, send it out on the way to FedEx or something. But they have been done, all the tests they could do to make sure they are safe to work with in the normal lab setup.
Okay, great. Annabelle, I'm going to ask you this one, because I think you said you were interested in genetics. How do you become a genetics scientist?
Well, I did my undergraduate degree in genetics. So I went to a university that offered that. But if dependent on kind of what life stage this person is at, I always took triple science at GCSE. That is a thing anymore. And then I did biology, chemistry, maths at A-level. And then I applied to universities with that course. So I went to the University of Glasgow, And pretty much as soon as you're undergrad, I think you can consider yourself a geneticist. But after that, there's plenty of research opportunities out there. So look for projects that involve anything with gene expression or recombination or anything to do with that.
Okay, so we're slowly running out of time. It's probably going to be the last question. So what's the best thing? It's a good question to end upon. What's the best thing about working at the LMB?
Right. I think I think it's great. So especially what we do is that we get to play the most state-of-the-art equipment that we don't usually have in other research institutes, and we have really fancy, not fancy, but really like smart and people around here to help us, intellectually as well as like details that they can actually work with you, stick around with you, and to push the difficult challenges through. I think it's just people and the environment that makes it very special.
Yeah, I think Sun Long is right. There's a really open atmosphere here. We all have open plan labs, so you can always speak to anybody in the building if you have any questions. And there are a lot of experts in all sorts of different fields here. So even if you're not working in that field, there will always be someone there to help you out. So I think that's probably the best thing about work. I've spoken to a lot of interesting and smart people.
Yeah, to add to that, exactly, it's very collaborative. So, for example, Zunlong works on exactly the opposite end of where I work, and we do interact quite frequently, asking each other for advice or sharing the same equipment. So it's a great place to work.
I agree.
I think sadly we've run out of time. Thank you so much for all your questions, even the tricky ones. And thank you to my lovely colleagues for answering them.
Thank you.
Bye.
Bye-bye. Bye.
So we've put a link in the chat for a short survey. If you could take a minute or so to complete it, we'd really appreciate it. So thanks again for joining us. And we've also had an absolutely fantastic time showing you around the Nobel Prize Factory. Now, all that's left for me to do is to show you out and make sure you don't get lost en route.